And here's the point about it is this, is that, and this is interesting because this is right out of the Science Journal, and this backbone here, this protein in green, is the same one that's in yellow. And in this case, this is a protein that causes muscle contraction in your body. And it depends on this signal. The signal is calcium. When calcium shows up, it plugs into the hole. It changes the charge, and it causes the protein to change its shape from this inactive form, conformation 1, shape 1. When I add the signal, it goes to shape 2, the active form. If I take the signal away, then the protein goes back to the resting state. So there are two different shapes to the protein, an active and an inactive form, and the activity is now controlled by the signal. So basically it says that proteins provide for your physical structure, but proteins also provide for your behavior. Your behavior is the movement, the actions that you express in your life, and the movement comes from the movement of protein. So basically it says this, your behavior represents the action of a protein that interacts with a signal. And so that the signal activates the protein to move, and the movement generates behavior. Well, why this is important then, it says this, if I have just the protein and no signal, what happens? Nothing. So then action is really by what? Controlling the signal. So here's the point. The brain of the cell is the structure that controls the signals to tell the cell what to do in response to the environment. So we want to understand the brain of the cell because there's a very limited time. And over the weekend, when I have 12 hours or so to talk about it, I can expand on it. But in a very brief moment, the brain of the cell is the skin of the cell, the membrane. It's the same as your skin. And you might say to yourself, what do you mean, the skin and the brain, they look like two different things? And the answer is this. In embryology, there are germ layers. There are 3 germ layers that create the ultimate full-size organism. The germ layers are called ectoderm, mesoderm, and endoderm. They're layers of cells. Each layer gives rise to different organs and tissues. Interesting, the outer layer, called the ectoderm, only gives rise to 2 things in the human body. Skin and the brain and nervous system. Your brain is derived from your skin. And it makes sense as to why, because I'll show you, the skin is the interface between the environment and the cytoplasm. The skin can read what's going on and then tell the proteins in the cell what to do. I told you I could take the nucleus out of the cell and didn't change the behavior. And the reason why is the nucleus is not the brain of the cell. The nucleus is the gonad of the cell. It's reproduction. If I need a part to make the cell work, then the nucleus is like the Butterick pattern drawer. It's got all the patterns to make 70,000 different parts in your body, but the nucleus doesn't know which one is needed at which time. The nucleus has no intelligence. The nucleus is just the repository for the pattern. So if I take the nucleus out of the cell, I didn't change anything about the cell. The cell will die after a while for the following reason. The proteins that make up the machinery break down and wear out. And if they break down, I got to replace them, otherwise I die. So I need the nucleus not for the intelligence, I only need the nucleus for the blueprint. So there's no brain involved with genes. Genes are not capable of that. How does the cell membrane work? It takes an environmental signal, and it could be anything. It could be sunshine, it could be hot air, it could be sound, it could be anything that's out there, chemicals, smells, taste, anything that's from the environment. The membrane picks that up, and the environmental signal is called the primary signal. And then what happens then is this, the membrane converts the environmental signal into the signal that controls the protein, so that the behavior is mediated by the cell membrane as it responds to the environment. The behavior, if I cut off the environment, the cell has no behavior. Your cell with all the components in it, if I could cut off the environment from the cell, it will just sit there. It has no life. Life is due to how the cell responds to the environment. Your life is how you respond to your environment. As you see the environment, as you walk out of here, those are environmental signals. They actually run your proteins and make you behave. So your behavior is not due to the genes. We didn't even bring DNA in yet. Your behavior is due to how you see the signal, which is called perception, and then convert that signal into selecting the right proteins for your responses. Okay? So now the issue is, how does this membrane work? Well, that's the beautiful part. It's relatively simple. Let me explain it. If we look at some cells growing on a petri dish, And we look at the skin of the cells. It's all bubbly looking, but at a higher magnification, this is what it looks like. At higher magnification, the skin is like a sandwich. It looks like a bread and butter sandwich, and there's this lipid layer right in the middle, and it's the oil that makes the skin a barrier because the water in the environment can't go through the membrane, and the water inside the cell can't go through the membrane. So under the skin, there's a protected environment of which all the mechanisms can work. To show you the reality of it, this is an electron microscope picture of the actual cell membrane. You see the dark light, dark layer of the cell membrane? It really represents the layer of these molecules that look like this, okay? Dark, light, dark, so that the model and the image of the real picture are very much exactly the same thing. But this membrane isn't functional because I left out the most important part of the membrane. It's the proteins that we were talking about that are capable of responding to the signal and then activating a behavior. So when I look at the surface of the cell, actually, instead of being smooth, there are all these structures like antennas sticking up all over the surface and proteins built into the membrane. And that these proteins read the environment and convert that environmental signal into a behavior. Let me explain how it works. Here's the membrane. Here are two different proteins. I'm going to tell you right now, there are thousands and thousands of proteins in the cell membrane, but I can divide them into two groups. One group has antennas on it. And antennas are receivers, just like your television antenna on top of your house before cable. When you had an antenna on the top, what was its function? To pick up a signal. And then what happened to that signal? It was transmitted down the wire to the television, and the television converted the signal into something you could see. Well, here's the point. Many of the receptors in the cell have these antennas sticking up from the cell. So if I go back, these are the antennas sticking up from the surface of the cell right here. And what they're tuned to are not television stations, they're tuned to environmental information. It might be glucose, for example. Is there sugar out there? Or histamine. Is there histamine out there which tells me to get ready for an emergency response? Or is there something like insulin which tells me to change my metabolic pathway? For every different thing the cell can see, it has a different antenna. So that means the cells are covered over the surface with antennas for everything the cell can deal with. So the signals come in and picked up by the antenna, but then they're converted into the behavior by the second class of proteins. There are three different kinds. Channels, which are just like olives with a hole in the middle of them where information can go in. Enzymes, which are proteins that cause metabolism to occur, or cytoskeletal proteins, proteins that change the shape of the cell. So if a signal comes in, let's say a toxic signal comes in, and this is a receiver for that, and it couples to the skeleton, it says, it's toxic signal, turn around and start running the other way, then this is the input and this is the output. So the proteins work in combination. Receptors receive signals. Do you have receptors? Yeah. What are the obvious ones? Eyes, ears, nose, taste, touch. Where are all your receptors located? In the skin. So you and the cell are parallels, but it doesn't look like an eye and it doesn't look like a nose, but its function is exactly the same. It's the equivalent of an eye. If this was tuned to light, a photon of light would hit this antenna and that cell would respond like an eye and say, I see the light. And once it sees the light, it has to convert that information, and it uses it by connecting to this, and I'm going to show you how that happens. So the input, the antenna, connects to the output, which makes the behavior. Let me explain how it works. So we'll use this model right here of a receptor. Let's see. This receptor is going to, I'm going to show it to you in dynamic motion. Here's my membrane, bread and butter sandwich. Here's my antenna, and this is the antenna scouring the environment for a signal. Look at the shape because the protein goes into the cell. Look at the shape. What happens when when a protein binds to a signal? Changes shape. Well, watch what happens. See, when the signal came in, and watch what happens when the signal goes away. Should I do that one more time so you can see that? Okay, basically it says this, is that the proteins are in the cell, that these receptors are migrating through the surface of the cell. Okay, here it is. Are you going to come start moving soon? Okay, here's the protein. It's scanning the environment, looking for signals. And let's say it's an insulin receptor. If there's insulin, I would activate the receptor. But look, no activation of the receptor. There's no change in shape. But when the insulin shows up, I change the shape. So if I'm inside the cell, if we were inside the cell and these proteins were hanging down from the ceiling, would you be able to tell if insulin was here or not by the shape of the protein? Yeah. So if you're inside the cell, you can tell what's going on outside. Okay? Now the next thing is this. After I receive a signal, I actually have to send a secondary signal into the cell to activate the behavior of the cell. So in this case, I'm going to use a channel, which is like an olive. And what happens is this. This protein moves around through the membrane. See, it's closed at the top. Nothing can get through. But when it opens, it makes a channel where signal molecules can shoot inside the cell. And the bottom line is this. The only thing that can go through this channel is what fits. If the molecule doesn't fit, it doesn't go through the channel. So basically it says this. In the resting state, the protein is closed. But when activated, the protein opens. And when it opens, it allows signals to go into the cell and coordinate the behavior of the cell. So now that we've looked at that, let's put the two together and then show you what is this mechanism that controls biology. The two together, the input and the output. This is looking for the environmental signal, and this is going to convert that signal into behavior. This is the connector. Look at the shape. Does it fit here, yes or no? Okay, so that means no signal is present. So no behavior. But when the signal shows up, I change the shape. When the shape is changed, then I connect it, open this one up, and see this? This is a signal that tells the cell to do something, and it's going to do whatever is connected to that. So the bottom line is the behavior of the cell is controlled by the input of the environment, and then the conversion of that input into a behavior signal that coordinates the function of the cell to respond to what's going on in the environment. Interesting point. If I cut the antenna off the cell so it cannot see anything, there's no behavior. The cell is totally comatose. It'll sit there, and what does that mean? It says then behavior is related to the signal. If there's no signal, there's no behavior. So it's like reflex, stimulus, response. The signal is the stimulus, and then the cell creates a response. So basically it goes like this. The receptor is the input. It reads the environment. The receptor then connects to the effector, which generates the behavior through a secondary signal. The primary signal, this is the secondary signal that activates the behavior of the cell. So that this unit is controlling a specific aspect of behavior. But there's like thousands of these different kinds of units simultaneously. So this is an example of just a switch. It's a switch. When insulin shows up, switch on metabolism. When histamine shows up, switch on protection. So for each different thing that's out in the environment, there's a switch that will activate the behavior of the cell. So basically, did you understand that as a switch, input-output? Well, then look at it this way. The function of the receptor is awareness of the environment, right? It sees. But I have to convert that signal into a biological action. So the function of the output guy, the effector, is to create a physical sensation or response to that signal. Is that understandable so far? Why is this important? Go back and ask, the first step is this. What does this do? What does it do? It controls a typical behavior in response to the signal. Well, what is this called? Well, it represents awareness of the environment through physical sensation. So what is this control unit actually called? Perception. Perception, awareness of the environment through physical sensation. So if I go back a second, I say, what is the function of this? And the answer is this. It's perception. It sees the environment and activates a behavior. There's no DNA involved. There's no genes involved. All we're talking about is stimulus response. Stimulus comes in the receptor, response made by the effector. Okay? So the point about it is this simple first conclusion. It's very basic. Behavior, which is movement, this is behavior. Movement of protein is controlled by the signal, but via perception. So the bottom line is this. Perception controls behavior. If there is no perception, there's no behavior. And all of a sudden it's like, oh, well, that's number one. We didn't have genes involved yet. So let's just see how this actually works. So basically, here are proteins in the cell. Let's say that these proteins carry out a specific function, respiration or digestion. We call it a pathway. So let's say it's a muscle contraction. That these proteins, when activated, what activates the protein? The signal activates the protein. So what we're going to do is convert an environmental signal using the receptor and the effector. And this time I'm using an enzyme. Okay? And what I'm going to show you is how the mechanism from the environment, the signal, well, here's my connector one. Remember the green guy? But now the signal comes, and when the signal from the environment binds to the receptor, it changes the shape or conformation of the receptor, which implies that the signal is there. So the protein informs the cell that something has happened. Now that that shape has changed, this processor protein, the green one, can bind to the shape because it wasn't able to bind before. It wasn't the right shape. It's like dominoes, one hitting the next. Now that the processor protein is bound to the receptor, it changes its shape and will conform to fit the enzyme. So one domino hits the next, hits the next. Now that this is connected, I activate the enzyme, and the enzyme is going to create a signal. Now here's the connection between my muscle proteins and the receptor. This is the secondary signal. Remember, primary signal and secondary signal. Well, at first it's bound and covered up and inhibited because if there's no signal, I don't want it to do anything. But when the signal shows up, then I take the enzyme, split it, and the active component, now this is the active signal, can bind to the protein. And now I can activate this protein. And this is the behavior that's going to be expressed by the cell. And the question, of course, is, well, what really activated this protein? What was the original source? The answer is the primary signal at the environment was then relayed by the secondary signal to activate the behavior of the cell. So without all the labels, if we just quickly look at it, let's just go right through it real fast. And the point about it is this. Let's see. That here's the environmental signal. Here's the receptor, the effector. That's the perception unit. Perception is now being started because it saw the environment, changed the shape, activates the enzyme. The enzyme is activated, breaks that molecule, the signal molecule. The signal molecule comes down, binds to the protein, and generates behavior. The bottom line was this. The behavior of the cell is not programmed. The behavior of the cell is continuously adjusting to whatever the signals are in the environment. So now I got another question to ask you. What happens if that environmental signal shows up, but I don't have the proteins necessary in the cell right now for that event? So when it shows up, it says, oh, I can't make a response. I don't have the behavioral proteins. Where do we get the behavioral proteins? Now we bring the DNA back in. What's the role of the DNA? The DNA double helix actually is a blueprint of the protein. If I separate the helix into two separate strands, and you look at these are called bases. These are the steps of the double helix right here. The color sequence in the DNA codes for the sequence of the amino acids. So for every 3 bases, I can tell you which is the next amino acid. So the point is this. The plan of how to make a protein, a specific protein, is built into this DNA. So that every 3 bases say, oh, put in tryptamine. Okay, put in proline. Next one, put in alanine, whatever it is. So the sequence, the DNA is a blueprint for the protein. Okay? That's all it is. It doesn't have any action except when I need it. So how do I activate the gene? Well, you heard of cancer genes? And you say, what is that, a gene that gives you cancer? And the answer is, here's a simple truth. Genes do not self-activate. That's biochemically a truth, meaning a gene cannot turn itself on and a gene cannot turn itself off. If you want a gene to be active, it's not up to the gene. So the concept that there's a cancer gene is a false concept, meaning this. If the gene really caused cancer and you were in possession of that gene, when would you express the cancer? You would express it by the time you were born. Because as soon as the cells started to divide, the cancer gene would say, okay, time to make cancer. So how can you have so-called cancer gene for 30 or 40 years sitting in your body and you don't have cancer and then you get cancer? Should I go back and say the gene caused that? And the answer is no. In this paper by Niehaut, Metaphors in the Roles of Genes in Development, he played it out in a simple statement of truth that I'm going to show you because I want to use his paper because the statement is so perfect. But the fact is this. What did he say? He said this. For 50 years, we have believed that genes are in control. We've been repeating it and saying it over and over again for 50 years so that it's part of every textbook. And the bottom line was that was never a scientific reality. It was never scientifically established that genes control anything. It's not true. What is the truth? Well, the answer is this. The first thing, conventional belief, genes control biology, is totally false. Why? Genes can't turn themselves on. Genes can't turn themselves off. How are they going to control anything? They can't control themselves. So bottom line is the genes aren't in charge. So the question is, if I need a gene to be activated, and why would a gene be activated? To make the proteins for the cell that needs to do the behavior. So the truth statement is this. When a gene product is needed, a signal from its environment, not an emergent property of the gene itself, activates expression of the gene. Well, that's somewhat of a complicated sentence, so let's simplify it. Just read line 2 and line 4. And if we read that, it says, a signal from its environment activates expression of the gene. What does that mean? The genes in your body are selected not because they're self-selecting. The genes are always selected in response to the environment that you're in. So if you had that cancer gene and for 35 years, let's say, you stood around saying, hey, I don't have cancer, and all of a sudden cancer happened, are we going to go to the gene and blame the gene, or what would we actually look for as responsible if you understand the true statement? The signal from the environment. What change in your life promoted activation of the gene that was sitting dormant for 35 years? We've been focusing on the gene all the time. The point is, we have to start focusing on the signals.